Hello, everyone. So glad you're here with me. It is Deep Dive Wednesday. You should know what that means, but just in case, this is the day where we go deep when we're talking about coronavirus. We peel back the layers. We take a look at the science, the myths, the latest headlines, and everything you need to know about the situation right now. It does seem as if every day we get new details. In fact, I don't think that's in doubt. We do get new details, but sometimes it contradicts what we thought we knew and understood about this virus. Some other days it's just downright frightening. All of it is coming at us fast. It's coming at us in large amounts, and we want to use these days to bring the experts in so that we can really sift through it all and try and separate fact from fiction and answer the questions that you have and bring you the information that you need to know. So joining us once more is Professor Yanis Ben Amour. He joins us. He's from Columbia University. He's here for a second time to share his insights, to really drill down on some of the stuff that he wants you to be thinking about. But also, it's an opportunity for you to get writing. Send us your questions. We want to know what you want answered when it comes to the question of testing, when it comes to the question of vaccines, when it comes to issues around treatment. Start writing those questions. Send them in. We're going to get to all of it. We have a full show. We're going to spend the full hour with Professor Ben Amour. And so I want you to be with us for that. And I want you to be part of the conversation because your voice and what you need to know is critical for us to make our way through this. Let's get started. This fancy new graphic. So before we get to the professor, let's take a look at the numbers and see what's happening on the continent right now with regards to the pandemic. Let's look at total number of cases. There you see there on your screen, 70,505. Again, just for context, last week we just went past that 50,000 mark, and here we are Wednesday, and we're at 70,505. Of those, the active cases, 43,607, and the deaths recorded right now, 2,432. I always like to stress to you that these are confirmed cases. We're not doing enough testing. We don't know the full scale of the situation. So those are just confirmed cases on the continent. Those numbers coming by way of Johns Hopkins University. When you look at Northern Africa, and we take a moment to pause, in Algeria, you may recall that Algeria has just started producing their own rapid tests. This, as the country goes past the 6,000 mark in confirmed cases. Egypt, we continue to watch it very closely. Over 10,000 confirmed coronavirus cases there. In terms of deaths, 556 recorded at the moment. I want to turn my attention to sub-Saharan Africa, Nigeria and Ghana leading the way in West Africa. Ghana, of great concern, over 5,000 cases, 5,127. Deaths recorded 22. You may remember that Ghana just a few days ago came across or discovered that large cluster of cases in that fish factory in the coastal city of Tema. One person infecting 533 people. There you see it there, 5,000 cases plus in Ghana. In Nigeria, over 4,787. Deaths in Nigeria, 158. For the people, for my people, Sierra Leone, let me give you a check as to where we are in Sierra Leone. 338 cases currently, 19 deaths. If we move further across the continent, we have to keep a close eye on Tanzania. Mark Tanzania down, everyone. The U.S. Embassy there is warning that we could see an explosive increase in case numbers. because the country effectively is not disclosing data. It is moving as if nothing is happening. People are crowding areas. Everything is going on as normal. But these are not normal times. We aren't getting full transparency from the government. What we know at this stage is 509 cases in Tanzania, deaths 21. The US Embassy there warning that we could see an exponential growth of cases. South Sudan should also be on your radars. Currently, they have 194 cases, no deaths, but officials now confirming that they have discovered coronavirus in a couple of the camps there. A couple of the camps with displaced people, they have discovered coronavirus there. We're watching South Sudan closely to see what comes next. And finally, Lesotho, which we have been holding on to as that singular beacon of hope, which it hadn't disclosed, it hadn't recorded any confirmed cases. That has now come to an end. We're coming to terms with the fact that Lesotho has recorded its first confirmed case of coronavirus. That means it is everywhere on the continent. Every single country in Africa has now recorded confirmed cases of coronavirus. Total number, 70,505 confirmed cases. People are contracting this virus and they are dying from this virus. You just heard the number that I gave you there, 2,432 deaths. We want to take a moment to acknowledge, to remember some of those who have lost their lives in this pandemic. I want to tell you about a really disturbing story out of London. You may have read it, but we want to tell you a little bit more about it. This occurred where a woman working in a train station, in fact, it was a Victoria train station to be precise, she was assaulted. She was assaulted by a man who came up and spat at her and her colleague. He told them he had coronavirus. He spat at them. And they both developed the disease a few days later. And the woman you see on your screens, Bele Mujinga, was 47 years old. And she lost her life. She had an underlying respiratory condition, but she died after she was assaulted by this individual. The British Transport Police are now investigating. And our thoughts and prayers are with her family, with her loved ones. She leaves behind an 11-year-old daughter. Truly, truly sickening. Another individual that we want to pay tribute to, Bana Abdallah Ali, a man who was known for his love of basketball. In fact, he took it to Somalia. He fought very hard to get the game to Somalia. He was in hospital for about a month after contracting the virus, and then he died in London on April 28. He survived by his loving wife and children. He was 54 years old. We think it's absolutely important that we pay tribute to those we're losing in this pandemic and keep their families in our thoughts and prayers. We want to hear your thoughts. We want to hear what you think about the situation, what you are going through, wherever you are, whether you're in the continent, in the diaspora. Send us your thoughts. It's important that your voice is part of the conversation. That's why we're doing this show. And that's why it is perfectly suited to this medium, because you can communicate and be part of the conversation in real time. So send us your messages. Tell us what you think. Tell us what you think about the show. Tell us about how your government, wherever you are, is handling the situation. What you think matters. So all you have to do is right now in this show, be part of it. We have Professor Ben AMOR joining us to talk about everything that we know about this virus right now. Start sending your questions. But beyond your questions and your typed messages, send us videos. We want to see you. We want to hear from you. Send them to our Facebook page, the Home with Isha Sesay Facebook page. They don't have to be long videos. About 2 minutes, just say your name, where you're from, and just share with us what you're going through. Be part of this conversation. That's why we're doing this, to build a dialogue that involves not just academics and world leaders, but your voice is at the center of it. So we want to hear from you. Send your videos, send your messages to our Facebook page, or right now, start sending your messages so you can be part of today's Deep Dive Wednesday. All right. If you were with me at the top, you heard me say that every day more information comes out. By the time I get to bed in the evening, I'm trying to figure out what I should be holding on to, and where my questions are. And every day I go to bed with a longer list of questions. There is so much. We're living in a time of information overload, and I know that you are going through it just as I am. And so that's why we do Deep Dive Wednesday, where we bring an expert in to talk to us about the situation and what we need to know. Professor Yanis Ben-Amour is here with us from Columbia University. He joins us now from New York. Professor Yanis, Prof, welcome.
Thank you, Aisha. It's a pleasure to be back.
It is so good to have you with us. We need you now more than ever. We need people like you who are able to take in all of this information and really process it, synthesize it, and tell us what we need to know. I do want to start today with the question of diagnostics, because it is confusing to many of us. There are so many different tests, and yet we continue to hear how difficult it is to gain access to the tests. And there's just too much information to process. So let's start with the question of testing, and the most commonly discussed, the PCR test. What exactly is that?
Okay, so PCR test stands for polymerase chain reaction. So this is the test that is specifically for people who have active viral infection. What it's going to look for is traces of the virus inside your body. Okay, so the way that you will do a PCR test, or in this particular case, an RT-PCR test, I will explain the difference, is that it has to start with a nasal swab. This is the sample that is going to be used because the virus exists in the nasal area of the body. It can also exist in the saliva, but there's only one test at this point that has been developed using that sample. So nasal area, you collect the sample, and then what you're doing is you're going to amplify that virus using this technique called RT-PCR. What it does is basically it makes millions of copies of your virus, starting with the virus that you have collected from the sample, and that is then easily assessed by a machine. Okay, so when you are suspected of having an active infection, it's an RT-PCR test that your provider will be doing. You will be providing this nasal swab, and then depending on the manufacturer, depending on the machine that is used, you can have a result between 15 minutes and 3 days.
Okay, and that's a huge gap.
It's a huge gap.
Why is it? Why don't we have one standardized?
Because every company is using different, basically, protocols. And so there are machines that are able to amplify and give you a result in 15 minutes. So, for example, the machine Abbott ID Now, from sample collection to sample processing, 15 minutes. This machine apparently has a 15% false negative rate. So I think that it still needs a little bit of development.
Sure.
The reason why it can take up to 3 days is because not every provider has the machine within their facility. So, for example, if you live in New York City, you can go to healthcare providers that are at the corner of blocks. They will be getting the sample from you, and then they will ship it to another facility. So that will take a day, and then it's analyzed, and then the result comes back within 3 days. Yeah. So,
and what about in Africa? Generally speaking, in terms of time of return,
I would expect it to be the longer technology, the one that lasts 3 days. But there is a company that is widely marketed in Africa called Cepheid. They're actually known for their tuberculosis test. They developed what I would call an espresso style machine because it's automated. All you have to do is to load a cartridge with the sample into the machine, and in the case of COVID, within 45 minutes, the Cepheid machine will give you a result, whether it's positive or negative. Fully automated. All you have to do is to load the sample into the cartridge.
Now, you mentioned that one particular company with their testing device that had the high percentage of false negatives.
Abbott.
Yes, across the spectrum for PCR tests, how reliable are they?
Well, depending on the company. Okay, so there is a group in Geneva called FIND, Foundation for Innovative New Diagnostics, and they have a catalog of every single commercial test that is available, whether it is RT-PCR or the other ones that I will tell you about. At the latest count, there is close to 300 commercially available RT-PCR tests, and they are not all good. That's the reason why you have organizations like the FDA that will look at the results of the test and they will give you approval. What we are facing in the United States is that there was an urgency in getting some of these tests onto the market and close to the patient. And so the typical process was shortened. Instead of doing a full review for each of those tests, which takes months, the FDA is providing now emergency use approvals, or known as EUA. So most of the tests that you will find in the United States right now are under EUA, not full approval. In the context of Africa, each country has to either go by a European certification or the FDA certification and consider that it's okay. I hope they don't, that they add also a level of basically their own approval. India would do that, for example.
We talked about PCR. Let's talk about the antibody tests, because that's also in the mix, and help us understand the difference in where you'd be at in terms of infection for one versus the other.
Absolutely. So there is a critical difference between the RT-PCR and the antibody test. So the RT-PCR will tell you whether right now you have an active infection and whether you are possibly infectious. The antibody test does not do that. And the reason for that is because when you develop an infection, almost immediately, the virus starts to multiply in your body, and that is what the RT-PCR is able to detect. You do not immediately have an antibody response. Mm-hmm. So whenever you have an infectious agent coming into your body, whether it's a virus or a bacteria, your body is able to fight it, with the notable exception of HIV, because HIV destroys the immune response. But lo and behold, even in the case of malaria, in the case of tuberculosis, you will develop an antibody response, typically between 5 and 10 days. And so what those tests are doing is that they're looking for the presence of antibodies. If you were not exposed to the disease, you don't have a reason to have antibodies. We are not born with antibodies to every disease in the world. We develop them as we encounter them. This is how our body is such an incredible machine. And typically these antibodies, depending on the disease, they can stay for life, or they will be around for about 2, 3, 5 years, depending on the disease. So what the antibody test does, after 5 to 10 days, it will tell you whether you had a past infection. Okay. So it's important to know that if you start developing symptoms, that you do not immediately go to the antibody test, because you could be positive, but the antibody test will show you a negative. And for the right reason, is because you just have not had time to develop that antibody response. Mm-hmm,
mm-hmm. But the question of the presence of antibodies still doesn't necessarily guarantee lasting immunity, because this seems to be a question that we're all grappling with when it comes to this novel coronavirus. You may have antibodies, but does that mean you couldn't be infected again? There are questions about how much, the volume of antibodies, how durable they are.
That is a fantastic question, and unfortunately, the most astute scientist does not currently have the answer. And the reason for that is this is a new disease. It's been around for less than 6 months, so we are still learning a lot. So this is as much as I can tell you. There is a study out of Mount Sinai, led by Florian Kramer, and what he's done is he has looked at the blood of people who had confirmed coronavirus. And in almost 98% of the cases, they had antibodies against the disease. So that's part of the equation. Okay. If almost everybody who has the disease develops antibodies. But the flip side is that not all antibodies will protect you in case you get exposed again. So what we need to now see with more patients and over a longer period of time, probably over 12 months, is whether reinfection occurs. So what scientists like Florian Kramer at Mount Sinai are doing is that they are actually analyzing the specific antibodies person to person, and they are going to look at ratios. And there's a particular antibody that we're interested in called neutralizing antibody. Mm-hmm. And if this is present, bingo, that means that you are protected against a further a reinfection. So the other piece of information that I wanted to share with your viewers is that, so COVID-19 is caused by a virus named SARS-CoV-2. Mm-hmm. And it was named SARS-CoV-2 because it is very close to another pathogen, another coronavirus that afflicted mankind back in 2002, 2003, called SARS-CoV. And that is the pathogen that caused the severe acute respiratory syndrome. Yeah. Now, because that disease happened 17 years ago, we have longer time information. Now, SARS, the original disease, only infected about 8,000 people, and only about 800 people died. Mm-hmm. The magnitude is much, much different than what we're facing now with this coronavirus, SARS-CoV-2, causing COVID-19. But the good news is that the genetic similarity between SARS-CoV and SARS-CoV-2 is about 73%. Mm-hmm. And when they analyzed the immune response of some of the people who had SARS back in 2002, 100% of them were immune for about 2 to 3 years, and some of them were immune up to 6 years. So that's some positive information.
Okay, I'll take it. I'll take the bright rays where I can find them. What's the difference between the antibody test and what's antigen testing?
Okay, so the antibody is the response that our body does against what we call an antigen. An antigen is any foreign body that comes into our body to invade it. So an antigen test would be a test that, for example, detects proteins of coronavirus. And there are a few now that are on the market. I believe there is probably only one that has received EUA, so this emergency use approval from the FDA in the United States. It is still based out of nasal swab, but it does not detect the virus directly. It detects the protein, one of the proteins of the virus.
Typically, why is this useful versus the PCR test?
Because it's faster. Because you don't rely on it has a flip side, unfortunately. So because it directly detects the protein, you don't have this amplification stage where you have to go from a few versions of the virus to millions of copies that can then be detected by the machine. This immediately detects it. But the flip side is that the sensitivity is lower, meaning that the number of times that it will tell you that a positive is lower than the PCR test. But it has its advantages because it is most likely to be point of care. So you can go to any facility. You don't require necessarily expensive equipment, and you are able to give some kind of a response.
So, I mean, right now, with all of these tests that you're talking about, whether it's the PCR, whether it's the antibody or the antigen, do people on the continent have to go to a healthcare provider? Where are we at the stage yet anywhere where, like with the home pregnancy test or the HIV test that you can now do at home? I mean, where are we in terms of access to these tests?
Yeah, that's a very important question. And unfortunately, Aisha, the answer is that there isn't a home test that is of good quality. And I think that the viewers need to know that, because unfortunately, you will find on Amazon and you will find online companies that market themselves as the home test with high sensitivity and high specificity, but these tests are not good. And so they can give you either a false positive or a false negative. And in both cases, it's not what you would want. Actually, the United Kingdom back in mid-March learned their lesson because they wanted to issue what they called immunity passports, with the idea that if you had the disease once, you were probably less likely to get infected again. And so you could go out and reopen the economy. And the best way to do that is to send these home tests so that people could do the test find out the result, and then either stay home if they were negative or restart the engine of the economy if they were positive. Unfortunately, they bought hundreds of thousands of tests, and the company that they picked was not good. And so they could not use them.
And I do just want to pick up on the question of immunity, just to get you to double down on it. You could gain immunity. I mean, we saw that with the original SARS, as you made the point. People had immunity. We could be looking at up to 6 years, at least in that case. But that doesn't take into account, or correct me if I'm wrong, that doesn't take into account the virus mutating, right?
Absolutely. So right now, it seems that this particular coronavirus has actually a very low frequency of mutation. And it makes sense from an evolutionary perspective. Here is why. We are all called naive, meaning we have never been exposed, and a lot of us don't have immunity. So there isn't really a reason for the virus to mutate, because it doesn't have an immunity that counters it when it gets into most people. Okay. Later on, when either we vaccinate or when most of the people had it, hopefully didn't die and develop an immunity, then it could be different. The virus will then, from an evolutionary perspective, have to mutate in order to infect more people, because a lot of people will have developed an immunity against it, and it will not be able to spread if it doesn't.
It's an intelligent enemy. You know, that's the thing about it. It doesn't respect borders. It's looking for body. It's inert until it finds a body, and then, you know, it replicates and it jumps. It's incredible, actually. Viruses are incredible. You know, I don't want to sound like I kind of am.
Yes.
Just to hear you say that, it will mutate. It will find a workaround, effectively, when it finds the obstacle, which would be immunity, or mass immunity, or herd immunity, or whatever. It will find the workaround and mutate.
So okay, I just want to tone this down a little bit, because it's not. There isn't an intelligent design. It's not that when we humans, with our brains and our capacity to think, when we face adversity, we can adapt because we understand the challenge and we can try to go around it. Viruses and bacteria don't think, oh, I am about to enter a human that has immunity against me, so I have to mutate. That's not how that works.
Well, how does it work?
So mutations happen all the time. Mutations are the reason why there was evolution to begin with. They are the reason why, unfortunately, some of us will develop cancer. Mutations, they are part of nature. And in the context of viruses and bacteria, it's how they adapt to the environment. Typically, when viruses multiply, in the process of multiplying, there are transcription errors that naturally happen. Most of these transcription errors are not of an advantage to the virus, and that new virus will just die out compared to the mother strain. But in the context of, for example, bacteria against which you have developed an antibiotic, let's say, for example, tuberculosis, we have antibiotics against tuberculosis, but in the process of multiplying, certain bacteria mutate and basically acquire, not because they want to, just by luck.
Okay.
They
so
acquire a resistance. It is not.
It's not triggered. That's the key thing.
It is not triggered.
Okay.
It is complete coincidence, and it is because it is a fight between the immunity and the virus, or the immunity and the bacteria, and you have this process of mutation that happens continuously. And when the immunity challenges, maybe one version of the bacteria or one version of the virus is now no longer being challenged by this immunity. As a result, that one will go through and start to multiply. That's how we have resistant bacteria, and they become a problem.
So interesting. Such an important point, that it's not triggered. It's a coincidence. Isha Sesay sent this question in. He says the genetic similarities between SARS and SARS-CoV-2 is 73%. This is an interesting empirical result. Does it mean earlier vaccines used for SARS need slight modifications to get an ideal vaccine for COVID-19?
Fantastic question. And I wish I could say we had those vaccines. Unfortunately, this is what happened. 2002, 2003, the world is trembling at this possibility of a worldwide pandemic, but then it didn't actually reach the level that it reached now with COVID-19. Like I said, only 8,000 people were infected and 800 people died. There is a massive difference between SARS and this coronavirus. It is about when people are infectious and when they present symptoms. SARS, the original one, had a very high mortality rate, about 35%, very, very high. But when people were infectious, you would see it. They were always symptomatic. So that made detection quite easy, but also placing people in quarantine. Mm-hmm. With this coronavirus, we know that the major issue is that there are many people around that are spreading the disease and they don't even know it. It's because they become infectious up to 48 hours before they are symptomatic. And some people are completely asymptomatic. Okay. So in other words, let's say today I start to have symptoms. And for the last 48 hours, I've been going around town and I was completely fine, no fever, no cough, no loss of smell. But I may already have been infectious, meaning that if I didn't cover my cough, if I touched surfaces, I may have shed virus that could have infected other people. SARS, the original one, didn't do that. When people were showing symptoms, that's when they were infectious and it was possible to quarantine them. Okay, so unfortunately, the initial scare against SARS #1 went away when the pandemic went away. So all of the labs who were working on a vaccine stopped working on the vaccine. And so as a result, we don't have a vaccine against the original SARS, which we could repurpose. And I want to add another thing. Okay. It's not obvious that immunity to SARS would give you immunity to this coronavirus. And there are examples. Because that 20% difference could still matter. There are examples of people who were infected with SARS, survived, and now got infected with coronavirus. And one of my colleagues at Columbia, his name is Dr. Ian Lipkin. He's called the virus hunter because he goes after those viruses whenever he had both diseases. So being infected with one did not prevent him from getting infected from the other one.
I do want to bring this question from Bev Archibald. She asks, what about children? Is the testing different? Bev, thank you for the question.
Thank God it is not different. You would do exactly the same. So if you are looking for the live virus, really the difficult step is collecting the sample. It is very uncomfortable because it's not taking a swab from the nose. It is the laryngeal area in the back. So you have to take this long cotton swab looking thing and go as far as you can and start collecting the sample. It is very uncomfortable. And with children, it could be a little bit difficult if the child is not staying steady. So in the United States right now, and most places will soon have that too, we're doing self-swab. So it's no longer a healthcare provider who does that to you and sometimes can go in a little too far. You are given the swab and you are told how to do a self-extraction of the sample. It's actually a lot better because you feel your own pain, which the healthcare provider would not necessarily know. But the test is exactly the same. There is no difference.
Okay, but I did want to ask, with it being the self-test, are you always guaranteed? I mean, I guess that the answer is not necessarily, but in terms of capturing enough of the virus to make the test.
So I don't want to confuse you or the viewers. Self-test does not mean at home. Self-test means only that you are in the doctor's office, and instead of having them do it to you uncomfortably, they will provide you with the swab, and under their supervision, you are going to do it. So they will monitor that you're actually staying in there long enough and that you are scratching, so to speak, long enough to collect the samples. And you're supposed to go into both nostrils to multiply the chances of collecting virus if there is virus.
Okay, good point to highlight. Let's talk about treatment. Take us through what we know. I mean, when this just started, at least it feels like when it just started, there was a lot of talk about hydroxychloroquine. Certainly, President Trump, seemed to champion it and said, "What have you got to lose?" Where are we now? Because that talk seems to have died down after some trials, small trials, but they seem to give some important information.
So before I answer, let me just preface by saying, unfortunately, there is no treatment for coronavirus right now. I want your viewers to know that, because that is important information in case they go onto Facebook or they're being shared information that there is a cure. There isn't. Okay. As of today, May 13, there is no effective treatment. Okay, hydroxychloroquine, it all started when a small trial in the south of France, in Marseille, around March, showed that with 20 patients, providing hydroxychloroquine, which is actually an anti-malarial drug, seemed to show effectiveness. It was a very small study, and it was not done in the way that you would do studies around drugs, which would be randomized controlled trials, which basically would split your patients into categories. Some of them get the drugs, some of them don't get the drugs, and then you compare. So there was no control, and the sample size was very small. Now, just to summarize, more and more and more evidence is coming out that not only does hydroxychloroquine not change the outcome of your disease progression, it could actually kill you. So the latest piece of evidence, and probably the largest one, comes from my own institution, Columbia University. My colleague, Dr. Neil Schluger, retroactively looked at 1,400 patient records, people who had used hydroxychloroquine, about 800 who had used, about 600 who had not been given hydroxychloroquine, and there was no difference in disease progression. However, more people in the group that received hydroxychloroquine ended up dying of heart complications. So across Africa, I hear, or I know, that because of the wide availability of hydroxychloroquine, since it is an anti-malarial drug, that it has become the first-line treatment. And I would urge basically the policymakers to reconsider that now, in light of these new publications that are coming out that are showing that for hospitalized patients, it does not change the outcome, and it could actually kill your patients.
Wow. Stark words indeed. Important for everybody to hear that clearly, and for policymakers to look at the data and the evidence that we have right now, which doesn't point to any efficacy of hydroxychloroquine fighting coronavirus. Remdesivir, made by Gilead Science, which again made headlines recently. First of all, just give me your opinion on what the trial showed, and is this, again, there's no treatment, but they say it helps in terms of speeding up, I guess, the recovery. Talk to me about how you're interpreting the data that's been released.
Well, Aisha, when you have no other option, anything that is slightly better than not giving anything, we'll take it. I think this is literally where we're at. I just want to give your viewers a little bit of a background. So remdesivir was actually developed back in 2015 to combat Ebola during the Ebola outbreak of Sierra Leone, Liberia, and Guinea. But the drug, even though it seemed to show efficacy, actually was never commercially available because it hit the market after the end of the pandemic. So now with coronavirus, or with COVID-19, they wanted to see whether remdesivir had, since it's also an antiviral, it had an activity against coronavirus. So I have to say that the progression of the finding is a little interesting, because I am talking about coronavirus almost on a daily basis, and it seems that if you hear what I had to say about it 2 weeks ago, it's slightly different from what I will tell you now, and it is because more evidence seems to be coming out. So 2 weeks ago, there was a study in China that seemed to indicate that there was, in fact, not really an impact of using remdesivir on COVID-19 patients. Then, last week, a large study in the United States seemed to show that, as you mentioned, Aisha, it actually reduces the duration of hospitalization by a few days, which actually makes a lot of difference. So, like I said, because we are looking for good news, it is not a cure. You are not going to clear the viral infection by taking remdesivir, but it shortens your hospitalization time, which is a success. So as a result, now in the United States, it is recommended. And you will see that many countries around the world are now precipitously trying to get approval for this drug in order to import it and to use it for their patients. Remember, 2015, it was never commercially available. So none of the countries made the process of approving it for local use in their country. So you would not be able to import it to India or to Brazil or to France.
Just so I'm clear, it shortens hospital stays, right? So I guess my question is, what is the thinking now in terms of medical practitioners, in terms of doctors? At what point do they feel remdesivir is the right way to go? Do you have to be severely ill, or I mean, when is it administered? How sick do you have to be for them to say it's time for remdesivir, or do you just turn up and you have a positive test and they say let's give you remdesivir?
No, it seems from the evidence that we have right now that it is when you are getting hospitalized, but you're not yet on a respirator, because even when you are hospitalized, there is unfortunately a progression, and being placed on a ventilator is the last of the stages. So people who are moderate to severe but not yet on a respirator, that's when you would have the biggest impact. But again, even though it's an antiviral, it does not clear the infection. So we are still looking for that drug that will destroy the virus. And so we don't have to get to that. Until we find it, remdesivir right now seems to be the best tool we have in terms of drugs.
Okay, there are questions here. I do want to get this one in because this is important. Not necessarily directly tied to remdesivir, but I think we should get to it immediately. Jennifer M.Y. Tengbe said, I had the virus and I'm now negative, but my mum and daughter are still positive but stable. We live together. Is there any possibility that I will be reinfected?
I wish I had the answer, and it's unclear right now. I'm going to assume that the answer is no. But what I would urge this viewer is make sure that you protect yourself as if you were negative, because we just don't know the answer right now. So you can, in the back of your mind, feel that you have a little bit of protection, but you should still act as if you were negative and do all of the gestures that will protect you. Wash your hands, make sure that they are with their faces covered, do the social distancing, open windows so that there is a circulation of air, do not take risks. You could be protected, but we don't know yet. So just put all of the chances on your side. And I am glad that you survived the infection.
Very much so. Jennifer, thank you for asking that question and for sharing that. Abdullah Sesay, another Sesay, there are a lot of us, Prof, just to warn you, says, how can two people who tested positive at the same time and after 14 days, one of them is now negative while the other is still positive, but with no symptoms at all?
Okay, so very good question. It has to do with the fact that some bodies are actually very good at clearing infections because their immune system is a little bit stronger than another. This will explain if you take 1,000 people and 1,000 people who had coronavirus and who succeeded in fighting the infection, and you start analyzing the antibody response, you will probably have 1,000 different responses. So for what we know from Mount Sinai, everybody develops antibodies, but not all of them might be protecting you against another infection. We'll find that information out. But what we know is some immune systems are just stronger than others. And so you are able to clear the viral infection faster than somebody else. Eventually, if you survive, you will clear that infection as well, the other person.
Abdullah, thank you so much for that question. Jennifer did have another question that I think is worth asking, because I know it's on the mind of many people. what does the next phase of this look like to you right now? The next phase of this pandemic?
Well, right now, as of April 1st, so a month ago, 93% of the world population was under some form of lockdown. This was necessary from a public health perspective, but as a society, as mankind, we cannot live like this. And the economy obviously has to restart because a lot of people rely on daily wages. So the next step is to marry public health knowledge with the economic necessity to reopen countries. So it is important that this happens in sequential phases. You cannot just switch on the light. Basically, we close everything off in one switch, but we cannot reopen everything in one switch. It has to be done in a sequential way because you don't want to find yourself back where we were March 1st, at least for the United States. Now, in the context of the African continent, they are 3 weeks behind in terms of the infection. So when you reopen, you don't want to find yourself the day before you closed, meaning that you could actually risk another outbreak. And from what we know historically, the second waves can be worse than the first one.
Yeah. Prof, I want to ask you about COVID organics. MADAGASCAR'S homemade tonic that President Rajoelina is out, pushing it out into the world, and says that it has curative and preventive properties. That again, I want most evidence, scientific data to support that. Can you please and tell me your thoughts on this as different African countries take in their batches?
Yeah. So let me start off by saying that I commend African initiative. You know, we are African. We want solutions to come from the motherland, but we have to be extremely careful about quack solutions that could actually endanger people's lives. So it's not clear to me what went into the thinking. What I can anticipate is that COVID organic is made with a plant that has antimalarial properties.
That's right.
And so you can see the thinking because of all of this message battering of hydroxychloroquine and hydroxychloroquine being an antimalarial. Oh, why don't we use a plant that is African and a product that is African made to fight this scourge? But hydroxychloroquine isn't working, so we don't know. I don't want to shut it down. It would be a dream if it worked. And if you look at WHO's response, interestingly, they also didn't shut it down. What they said is, listen, before you go out and advertise it, before you say it is the cure, we need to at least do some research and confirm. If your evidence is that 2 people took it and they didn't die, that is not evidence. It is not scientific evidence that it's working. So if we want to have an actual answer, let's do a trial and compare people who take it and people who don't take it and see whether it works. Let me tell you, I don't want to dismiss it. In the fight against malaria, there was a story that some people out of China were chewing on the tree of the bark of a tree and were never getting sick. And that's how we discovered artemisinin.
Which is what's in this COVID organics.
So if you remember the story of aspirin, it's the same way. Women have menstrual cramps once a month. In Germany, there is a pharmacist who noticed that women who were chewing on the bark of a specific tree were never having these cramps. So he analyzed what was in the bark and he discovered aspirin. So it is possible that there are natural remedies that will work. And that's why I don't want to dismiss this as a possibility. Right now, however, the evidence does not seem to show that it's working. So let's do tests and let's show that it's working before we go and we advertise it and we sell it to other African countries.
Agreed. So well said. Convalescent plasma. as we talk about treatments as well. Where are we in the development of that? Where are we in terms of its efficacy in treating COVID-19?
Okay, so let me introduce the concept of convalescent plasma. So the idea there is that if you had COVID-19 and you successfully fought it, you now have antibodies, and we are able to harvest those antibodies by a blood donation and getting the plasma. And then we would put this plasma into a person who is currently fighting COVID, and it would help them. So I know that some of your viewers will have questions about some of the things that I said before, and I will get to that. So the idea is that you have antibodies because you survived, and so now you are transferring these antibodies. FDA recommends it. There are many clinical trials now going on in the US. One of the large ones is in New York at Mount Sinai. The NIH is sponsoring one as well. And the idea would be wonderful. It is the best of humanity. You had the disease, you survived. Now you want to do something to help somebody else. So you go, you give blood, they harvest the antibodies, and they give it to somebody else. It seems to be working. So to go back, if it seems to be working, does it mean that we have immunity? Well, probably. But the question is, would every convalescent plasma be working? And we don't know that yet. So what are they doing? They are actually taking convalescent plasma of people who had severe coronavirus, COVID-19. So people who were hospitalized, for example, or people who were on a respirator and survived. So that blood would have a higher immunity and is more likely to actually protect somebody else. It is my dream that, of course, anybody who survived any infection, even asymptomatically, would be able to go and provide blood.
Yeah.
I should say in the United States, that's not true. There are restrictions on who can give blood.
That's right.
That is a whole other topic.
That is a whole other topic. I do want to, because we have about 10 minutes left, I want to talk about vaccines. Can we talk about vaccines and where things stand in that area?
Sure. So it's very encouraging that for the first time in mankind's history, everybody is coming together. Researchers from competing labs are working together to try to find a solution against this scourge. What I can tell you, last time I checked, there were 119 candidates at various stages. Mostly are in preclinical, meaning that we are developing either the attenuated virus or a protein or something that we will inject into the human to create this vaccination. 8 of them are either in phase 1 or in phase 2. Actually, I can tell you 7 of them are in phase 1. 1 of them is in phase 2. What does that mean? So phase 1 is when you are looking whether this vaccine is harmful to people who are healthy. So typically phase 1, you would work with 40, 50, 100 people, and then you would vaccinate them. So you would inject them with this vaccine, and then people who are healthy, you see whether they develop side effects. Does it have an impact on their heart? Does it have an impact on their liver? And so you have to follow that. If it doesn't, you move on to a phase 2, which is a much larger trial, but still with healthy people. There is one candidate coming out of Oxford University that seems to be doing quite well. So Oxford University, in every interview that they're giving, are saying that they are 98% sure that by September, they will have a vaccine that works. And all of the data that they are producing right now seems to be showing that that is true. But there is a problem. If the vaccine is developed, that doesn't mean they will be available. And yes, it will depend on how quickly we are able to manufacture it at scale to vaccinate 7+ billion people, not just people in the US and people in Europe. We need to vaccinate everyone. And so that means we need to have enough doses for the entire population. Hopefully it's just one and not multiple. That would be another problem. So it is a problem of manufacturing. It is a problem of supply chain. It is a problem of delivery, and it will be a problem of cost. Because if this vaccine costs too much, you will see again our motherland, the African continent, may have a problem getting procurement. So there are a few candidates in the United States that are going through clinical trial, and what they're doing is they're taking an enormous risk, is that they're actually mass producing the vaccine before they know that it's working.
Wow.
Because they want to be sure that the minute they show that it's efficacious, that they're able to distribute it. Some of them will not be able to do that because the clinical trial will show that it doesn't work or that it has side effects, but they are taking this risk because they want it to go out fast. Oxford is not one of them because they are a university and they don't have a manufacturing, as far as I know, they don't yet have a manufacturing partner.
Wow. I do want to ask something very quickly because we're running out of time. Do you have any concerns that the anti-vax movement, anti-vaccination movement, will prove to be a major problem in terms of getting people to take this vaccine, especially in this age of social media when we've seen so much bad information floating around? How concerned are you about that?
I'm very concerned. These are people who survived child diseases and child mortality because they were vaccinated by their parents. And now, for, let me call it bohemian reasons, they have decided that they don't want to do that. Okay, let me try to justify something. There was a paper in a famous journal called Lancet that seemed to associate vaccination with autism. That paper has been withdrawn because the science behind it was shown to be wrong. Unfortunately, the message is still going around. So many concerned parents are worried about injecting to their babies something that could potentially harm them. So this is where it originated. So yes, I am very concerned. We hear so many stories about outbreaks in different communities in New York, in Europe, of people who do not vaccinate, and all of a sudden you have measles again. So there has to be a political effort. I mean, the politicians understand the economic consequences. If the politicians are assured, and that is important, if the politicians are assured that it is safe, because the clinical trials will have shown it is safe, then they have to take measures to impose vaccination. So in Italy, for example, some bohemian parents did not want to vaccinate their children. They were not allowed to send them to school. Mm-hmm. And I think that is it could have negative consequences for the child. But if the parents are concerned about their children, they will do the right thing.
Professor Beno, thank you so much for this great conversation. One question, slight change of tone, but I think it's important, and viewer centered it. Ali Jallow said to ask you this, and it was a please, and I could feel the insistence. Please ask the doctor if domestic pets can transmit this disease.
So we don't think so. So the evidence show that it is possible for humans to infect pets, but not the other way around yet. There was a famous example in the Bronx Zoo in New York where a handler infected a tiger. The tiger developed coronavirus. We know that it's possible for humans to infect cats. I have not heard evidence with dogs, but it's possible to infect cats. I don't know whether it is not known yet whether in return cats can then infect other people. So we'll have to watch that space.
We will have to watch that and so much else. Everyone is asking, well, there was one question that also asked about, we'll send that question along to you, Professor, somebody asking about you. You have a fan. We'll send that along to you. But thank you. Look, he's grinning now. He doesn't know what I'm going on about, but we'll pass it on. Thank you so much for spending the hour with us and for providing such valuable information. I hope you'll come back again. We would love more of your time and more of your insights. So we'd love to do another session with you.
Thank you for giving me the opportunity to speak about coronavirus.
Thank you, Professor. Be safe.
You too. Thank you.
Thank you. Thank you so much to you for all the questions you sent in, for your active engagement throughout the hour. We're going to keep doing these. Every Wednesday is Deep Dive Wednesday. So you know that if it's Wednesday, we're going to have an expert. We're going to be digging deep into the story and seeing what it means for us as Africans, for the continent, Africans in the diaspora. We are going to do all of that for you every Wednesday. Tomorrow, we're back with a full show with guests and great conversation and the very latest on the pandemic. You will not want to miss my conversation with tomorrow's guest, with Professor WILFRED MBACHAM. He's not only a biomedical scientist who graduated from Harvard University, he's also a chief. He's also a chief. He's chief of Bomé, a village in Cameroon. So we've got a chief who is a biomedical scientist who graduated from Harvard. He's going to be on the show tomorrow talking to us about how he is spreading the word amongst those in his village and helping them stay safe from coronavirus. He will be on the show tomorrow. Be sure to join the conversation. Again, we want your questions. I want to hear your voice. I hope you enjoyed this hour. We will be here every day throughout this. We're going to keep broadcasting and keep bringing you what you need to know. Please stay safe, everyone. See you back here, same time, same place tomorrow. Bye for now.